Hello, good morning. Welcome to Lecture 15. Today's lecture is in between Unit 2 and Unit 3. It's really a standalone lecture, sort of like a bottle episode of your favorite television series, one that doesn't really advance the overall narrative of the course, but one that allows us to dive deeper into a particular subject area. And the goal of today's lecture is really twofold. One, it serves as a basic introduction to the primary literature as it relates to cell and molecular biology as a discipline. And the second thing that today's lecture accomplishes is it lets us dive a little bit deeper into Alzheimer's disease as a subject. And specifically, the topic of today's article is the cloning of the beta secretase enzyme, BACE. So the reading for today is the primary article that first described the sequence of the beta secretase enzyme. It represents the first molecular cloning of the cDNA that encodes for the protein that performs the beta secretase proteolytic activity. And the enzyme was named BACE by the authors of this paper. This paper is posted on Moodle, and please take a look at it. If you can, it might even be beneficial to take a look at it either before listening to the rest of this lecture or concurrently with the lecture. What I'll do is I will first introduce the topic at hand by way of sort of an introduction, and then we'll go through the figures of the paper. So a little bit of background first, and then we'll simply systematically go through the figures of the paper. And what I'm hoping to accomplish here is a couple things. First, I want to give you a sense of how scientists communicate new findings. So we're going to talk about the primary literature as a vehicle for the communication of new data and new discoveries. Second, I want to give you a sense of how one might take a research article and then turn it into a PowerPoint presentation for purposes of, discussing it with an audience, for example. That's an exercise that we do in a lot of our upper level courses in biology here at K, and it's something that I'd like you to be exposed to in this intermediate course. And I guess the last thing I'll mention is it really helps us dive into the way scientists approach the work, the way people go about trying to understand the molecular basis for a devastating disease like Alzheimer's. So a lot of cool features of today's lecture. I will more formally introduce our third unit in our next lecture, Lecture 16. So again, this one sort of serves as an in-between between Unit 2 and 3. And the real point of this is to give us a sense of how we would go about cloning an enzyme like the beta secretase. Before we jump into the paper itself, I want to spend some time setting the context. It's always important when we're telling a story about a scientific discovery. It's always important for us to set the context and talk about what the issue at hand is. So at the time this paper came out, we knew a lot about the proteolysis of APP. We knew that there were two competing proteolytic pathways, one involving a beta secretase enzyme followed by a gamma secretase enzyme that would produce the pathogenic amyloid beta fragment, and a competitive sort of alternative pathway involving alpha secretase and gamma secretase that would instead produce a non-pathogenic fragment called P3. So we knew about those activities. We even knew the molecular identity of two of the three secretase enzymes. Alpha secretase was known to be TACE. And you heard a little bit about this in our bioinformatics module in the lab, how these are actually the same enzyme. So scientists had been able to go in and clone the gene that encodes the alpha secretase enzyme, and they named it TACE. We also knew the molecular identity of gamma secretase. And this was known to be a protease called presenilin. The only one that we didn't know back in 1999, when this work first was published, was the molecular identity of the beta secretase. It was really the last of the three to be successfully cloned. So there was a race underway. There were multiple labs, three really prominent labs in the world, two here in the United States, one in Ireland, that were working very hard and really racing with each other to be the first group to clone the beta secretase, to clone the gene responsible for that activity. So that's really the context in which this paper emerged back in late 1999. So here's a little capture of the paper itself, of the title of the paper. So I'll just walk through it. The journal this was published in was Science. Science is a very prominent weekly journal. It's very broad. It includes cell and molecular biology. It runs the gamut of biology all the way up through ecology. It also includes articles from physics, from chemistry, geology, you name it. So it's a very broad journal, very prestigious one to publish in. This paper is from October of 1999. And here is the author list. We can get a sense of the number of people it took to produce this work. And all of these authors, they come from mostly private industry. Amgen is a company that's still around in Thousand Oaks, California, and it's really an excellent biotech company. And then a couple of authors worked at Harvard at the Department of Neurology and the Center for Neurological Diseases. And the title of the paper you can see here, Beta Secretase Cleavage of Alzheimer's Amyloid Precursor Protein by the Transmembrane Aspartic Protease BACE. And what this is really saying is cleavage of APP by a membrane-anchored protease that they named BACE. Okay? A little bit more background to further set context. So we knew, of course, at the time this work was performed, that Alzheimer's was characterized by plaques that are extracellular and that those plaques contain amyloid beta. I showed you this immunohistochemistry micrograph in an earlier lecture. And what we're looking at here, just to review, is tissue from an Alzheimer's patient's brain that's been sectioned and stained with an antibody raised against A beta. And all the brown signal that we see, I'm circling a plaque here, here's another plaque, all this brown signal is extracellular and accumulated in soluble amyloid beta peptide. And remember, it came from the proteolysis of APP. So inside cells, APP was proteolyzed by beta and gamma secretase, and then the amyloid beta was secreted, it accumulated, and it formed these plaques that we can detect in the pathological specimen. So we knew a whole lot about the beta secretase activity, and I want to just make the point that knowing about the activity is not the same as having the cloned gene. We can describe the activity, we can look at the effects of it, we could even figure out where it happens in the cell and what the consequences of the activity are without knowing anything about the actual gene and the sequence of the gene that encodes for that activity. So they're very different things. But there's a limit to what we can learn by just describing this generic beta secretase enzyme activity. We really need to clone the gene so we can do specific analysis related to that enzyme. We could do mutagenesis to test whether different parts of the protein work in a particular predicted way. We could look at whether or not that gene is mutated in Alzheimer's disease cases. We could make a transgenic animal that would express that gene or make a knockout version. All those things are possible once we clone the cDNA. So these two are very, very different. And the goal always is to get the cDNA, so we have a lot more power and a lot more experiments to perform. Having said that, we still knew a lot related to the beta secretase activity. So let's take a look at what we knew and what some of the predicted requirements would have been. So first, because of the fact that amyloid beta is secreted and now collects in the extracellular space, it's predictable that APP is in the secretory pathway. And indeed, we know that. APP's gene had been cloned earlier. It's already been characterized by the time this paper comes out, and we know that APP is in the endomembrane system. It would make sense for beta secretase, the enzyme that proteolyzes APP, to also be in the endomembrane system. Otherwise, how is the substrate going to interact with the enzyme? Another prediction that we could easily make is that the beta secretase enzyme, whatever protein it ended up being, would have to be able to function in an acidic environment. And the reason for that is the endomembrane system organelles are acidic. And, the trans-Golgi network, for example, has a pH around 5. So any enzyme that works in that organelle would obviously have to be functional in an acidic environment. Any beta secretase candidate would have to be in the membrane, or at least would likely be in the membrane, because remember, APP, the substrate, is in the membrane. Okay? So it's much easier for APP to be proteolyzed by a protease that is also anchored in the membrane. So they would predict that their beta secretase was anchored in the membrane, and that would give it access to the beta cut site. And then finally, not finally, but continuing, we would expect the beta secretase enzyme to be expressed in the brain and to have broad expression. And here's the logic behind this prediction. I think the brain part is obvious. That's where the plaque is found. The brain is that's the organ in the body that's affected in Alzheimer's disease. It would be certainly weird if the beta secretase enzyme weren't expressed in the brain. It wouldn't be a very good candidate, would it? So we can easily predict, just with logic, that any beta secretase enzyme candidate would need to be expressed in the brain, and probably at pretty high levels. But intriguingly, amyloid beta is also made outside the brain, in the periphery, in other tissues. So we would also expect broad expression. Just as a side note, the amyloid beta produced in other tissues is not pathogenic. It doesn't seem to be really clumpy or sticky. There's something about the brain environment that's unique in the creation of an insoluble plaque. So, sort of an interesting observation and just simple logic that helps us to predict high expression in the brain and broad expression. These last two have to do with biochemistry, and also I think just really clear logic leads us to these predictions. If indeed your candidate beta secretase enzyme is a beta secretase, if you overexpress it, you ought to get more beta cleavage products. So overexpressing extra copies of the beta secretase enzyme ought to shift the competitive balance away from alpha secretase and elevate all of the beta and gamma cleavage products, including amyloid beta. So that's sort of like the opposite approach than the one we took in the lab. In the lab, we shifted the competitive balance in favor of alpha secretase. Well, if we did the opposite, we shifted the competitive balance in favor of beta. Instead of lowering amyloid beta like we found in the lab, we should expect that we would increase its levels. And these scientists are going to show us that. Last but not least, the pure test, or the really gold standard test, if you will, of an enzyme is if you put it and only it in a tube with its substrate, it's going to cleave. So they're going to test whether or not you stick their candidate beta secretase in a tube with APP and nothing else, make sure that that enzyme actually cuts where we think it will. So as we're going to see, Vassar et al., the authors of this paper, were able to essentially go through this entire checklist of requirements for beta secretase and check each one of these boxes as they walk through their paper. And I'll give you a sense of that as we move along. Anytime that we're describing and communicating a scientific paper, We want to first set context, as I've just done, and then we want to give a sense of the methods and the experimental model. We want to have a sense going into it of what the authors did. How were they able to demonstrate that they've cloned the beta secretase? So I want to talk to you here on this slide about how BACE, which is their cDNA and their protein that they isolated, was identified as a candidate. And it's a very, very clever approach. And I want to spend a little bit of time on this because it's one of the more elegant approaches that we'll see this term. So let's imagine the following scenario. Let's imagine that we have a cell line. It's a eukaryotic, immortalized cell line that we can culture in a dish. So there's the dish, there are the cells. They're happy and healthy. We've got them in the right culture media, etc. These cells are called 293Ts. Doesn't matter why they're called that. It's a weird name, I know, but that's what they're called, okay? And let's imagine that we take conditioned media from those cells, okay? So we just pipette off the media, and let's imagine that we perform an ELISA that allows us to measure amyloid beta. And we're just asking the question, is there amyloid beta present in the conditioned media of the 293T cells? And let's imagine that yes, indeed, there is. There's a whole lot of amyloid beta. Now, knowing that, and knowing nothing else about this cell line, we absolutely know, just with simple logic, that this cell line, these cells, must express beta secretase and gamma secretase. Somewhere in the genome of this cell line are the genes for these two enzymes, and not only are those genes present, they're expressed, right? So the beta secretase transcript is being turned into the beta secretase protein, same for the gamma secretase, and they're functional enzymes in these cells that allow us to produce A beta. Otherwise, how would the A beta have gotten into the conditioned media? There's no other way. It's not in the feed media that we're growing these cells in. The only way it gets there is if these cells make those proteins. So if you want to go fishing for a beta secretase cDNA, this would be a smart place to look. So how do you do that? How do you go fishing for the beta secretase gene sequence in the 293T cell line? Well, what you do is you generate a cDNA library. A cDNA library is a collection of all the expressed genes. So because you start with mRNA and turn it into cDNA, the fact that we're starting with this means that we know those genes are expressed. So we call this an expression library, and the approach that we're going to be talking about here is called expression cloning. Here's the way it works. We take this cDNA library, and for them, they had 860,000 cDNAs in their library. So 860,000 cDNAs that they made from the 293T cell line. How do they do that? They took those cells, they lysed them, they isolated RNA, they further isolated mRNA, they used reverse transcriptase to turn it into cDNA, and then they took each of those cDNA inserts and they put them into a vector that they could then use later for transfection. They ended up with 860,000 individual cDNAs. One of those 860,000 is the cDNA that encodes the beta secretase. Now we just have to find it. How the heck would we do that? Well, what we do is we take this and we dilute it or sort it, however you want to think about it, into pools of 100 cDNAs. So 8,600, I'm not going to draw 8,600 little aliquots here, but you get the idea. Aliquots of 100, okay? And then we're going to transfect this pool of 100 into the cell line. And then guess what we're going to look for? Well, we're going to look in conditioned media with ELISA for an elevation in amyloid beta over baseline. Isn't that clever? If our beta secretase cDNA is in this pool of 100, when we transfect it in, we should elevate the production of amyloid beta because now there are more copies of the beta secretase in those cells. Okay? So all we have to do is screen 8,600 pools, find the one pool of 100 that gives us the elevation in amyloid beta. Then we throw out all the other ones. And what do you think we do with this pool of 100? We dilute it further. So let's go into 10 pools of 10. You get the idea. And then we're going to transfect this pool of 10 in and look for that elevation in amyloid beta. And we throw out any of the ones that don't, and then we dilute this into individuals. And that's how we come about finding our cDNA candidate that we think is the beta secretase. It's very clever. Expression cloning. So what they did was they did this. They found their candidate clone. They sequenced it. And then they hypothesized the following. If they take this cDNA that they named BACE and they overexpress it, you should get more beta cleavage products. If you lower the levels of this, you should get decreases in beta secretase products. And that's just the biochemistry. Beyond that, all of those predicted requirements for beta secretase should be met by this candidate. And that is exactly what they hypothesized and exactly what they demonstrated. And at the end of it, what we have here is the molecular identity of the beta secretase activity, BACE. Okay, so we've given the context, we've given an experimental overview and a hypothesis. Now let's look at the data. And first, figure 1, we're going to begin with just a schematic that the authors have given us so we can better visualize what BACE, their candidate beta secretase, may be doing. So let's start here in the cartoon, and what we're looking at is the membrane. This would be the cytosol, and this would be the extracellular space or the lumen of the secretory pathway. So we're looking at the bilayer. Here's the inner leaflet, here's the outer leaflet, and we're looking at the substrate. Here's APP embedded within the membrane. The black box is the A beta sequence. Okay, so this would be the N terminus of APP. This would be the C terminus. And we see this big extracellular portion. And then we see the beta cut site, the alpha cut site, and the gamma cut site for amyloid beta. Now let's take this portion of the protein and let's blow it up. And let's look at the amino acid sequence within that part of the protein. And what we're looking at here is the beta secretase cut site. That's where beta secretase cuts. In between this methionine and this aspartic acid. Okay? So that's the cut site. That's where their candidate beta secretase clone, BACE, should cut. And later on, we'll see that it indeed does. Here, what we're looking at for this part of the cartoon are the proteolytic products of cleavage. And it's basically the same thing that I've drawn out for you a million times, and I've shown you the figure of APP proteolysis several times. It's just rendered a slightly different way. I'll help you walk through it. So full-length APP is cut by either alpha or beta secretase. Here's the alpha product, and here's the beta product. So here's the SAPP alpha fragment that gets secreted. Here's the SAPP beta fragment that's secreted after beta cleavage. And then here's the rest of the C-terminal fragment that remains after either alpha or beta cleavage. And then gamma cuts. And for this one, the product is P3, and for this one, the product is A beta. Here, they're showing us the sort of predicted domain architecture of their candidate beta secretase, BACE. They expect that it's also in the membrane, and that its active site that they're representing as kind of a star, I guess, is sticking into the lumen of the secretory pathway or outside the cell. And this is really important structurally because, well, why do you think? You tell me. If you guessed because that would put it in proximity to the beta cut site, then you're correct. So it wouldn't make sense for the active site to be in here because it would be on the other side of the membrane from the beta cut site. So that prediction we're also going to see is consistent with their data. All right, let's take a look at the data themselves. Beginning with sequence. Anytime you clone a cDNA, anytime you clone a gene, first thing you're going to do is sequence it. And you're going to take that sequence, that cDNA sequence, and have it translated into amino acid sequence. And then you're going to perform a blast analysis of that protein sequence, and you're going to see what the predicted structure function of your candidate, or of your cDNA protein rather, is. So here is the analysis for BACE. They're giving us the sequence in the single letter code. So you see, methionine, alanine, etc. And amino acid position 1 is here. And BACE is, as you can see, a 501 amino acid protein. So it's got 501 amino acids. And let's see what we can learn based on the sequence analysis. And let's actually begin here. Here's a stretch of 20 or so hydrophobic amino acids in what is a predicted transmembrane domain. So if you think back to that checklist, we said that the candidate beta secretase should be in the membrane. Check, it seems to be in the membrane. It has a TM domain. Okay, look at this. N-terminal stretch of hydrophobic amino acids and a predicted signal peptidase cut site right there. That looks a lot like the signal sequence that targets proteins to the secretory pathway, doesn't it? And remember, we know the substrate for BACE, APP, is in the secretory pathway. It makes sense that the enzyme would be as well. So that's another nice check that we get. See how powerful sequencing is? We just get all of that good information simply by sequencing and then making these structure function predictions. It looks like BACE is a zymogen because it has a prodomain, shown here in pink. Remember, prodomains are inhibitory. They usually block the active site of an enzyme. We got to proteolyze it away in order for the enzyme to be active, and that's where the cut site is, right there. So BACE is a zymogen, just like TACE is, by the way. That's kind of cool. And then we can look at other features. You can see that they've put a star above these asparagines every once in a while. These are predicted sites of glycosylation. Remember, N-linked glycosylation on asparagine happens in the ER. Again, driving home the point that this is a secretory pathway protein. They're showing us these cysteines with a little arrowhead. Remember, cysteines contain a sulfur in their functional group, in their R group, that are capable of disulfide bonding with other cysteines. So these are probably residues, amino acids within the protein that are important for folding. And then finally, look at this little box, DTGS, DSGT. Those are classic consensus motifs for aspartyl proteases. Aspartyl proteases have an aspartate, that D that you see in the sequence, in their active site, usually 2 of them in the protein, and that's exactly what we seem to have here. So from this, we learn it's in the membrane, it's in the secretory pathway, it's a zymogen, it's a protease, etc. Pretty cool. And now we can go about further describing this, but we do learn a lot just by looking at the sequence. So if BACE is the beta secretase, then its expression pattern should be what we would expect. It should conform to the expected pattern. So how do we look at expression? Well, we use our cDNA to generate a radio-labeled probe that we can then use for a northern blot. Remember that northern blotting is looking for mRNA. And remember that mRNA is a product of gene expression. So, DNA gets transcribed into mRNA. That means the gene's expressed. Okay? So here we're taking different parts of the body and we're looking in those tissues at whether or not we see BACE expressed. This is done in the mouse. So the library that they use, they cloned human BACE, and they're just using that to look at expression in a mouse. So they take a mouse, because of the conservation of sequence, your probe is going to work just great in a mouse. Mice and humans are pretty similar from a sequence perspective, remember? And so we take tissue, we extract RNA, we isolate mRNA, we run it on a gel, and here there are two gels shown here, one here and one here. The first gel has tissue from the pancreas, the kidney, the skeletal muscle, etc. And here is the signal. I'm going to show you the 3 bits of signal that we get. Every time we see this nice little black band, we know that we're looking at our probe hybridizing to mRNA in the northern blot. So one thing we learned from this is that BACE is alternatively spliced. We see these 3 different sizes of transcripts, so there's some alternative splicing that's been going on. Remember that the intensity of the signal directly relates to the amount of expression. So let's take a look here in the brain. We see pretty good expression of those. The pancreas actually has the best expression, oddly enough, and I'll tell you why that is in just a minute. And then you can see some other tissues that have relatively good expression of at least one of the alternative transcripts. Down here, if you're wondering what we're looking at here, we take this blot, we strip it of its probe for BACE, and then we probe it again for actin. Actin is what we call a loading control, and we should see relatively similar amounts of actin across all these tissues. And it helps us to know that the absence of signal here, for example, isn't because we simply don't have any RNA on that blot. So we know that there's RNA there, there's just not base RNA expressed at high levels, at least in the lung. Loading control. We often use cytoskeletal components for that. And if you're wondering why there's like a big band here for skeletal muscle and heart, well, that's muscle. And remember, heart or skeletal muscle is going to have a lot of actin because of its role in sliding filaments and contraction. So that's not surprising. Why is there more BACE in the pancreas than the brain? Later on, after this paper came out, we learned that BACE may be doing something totally different in the pancreas. It may be aiding in digestion. Turns out a lot of aspartyl proteases help to break down material in the pancreas for digestion. So BACE may be playing a role like that in the periphery. It's not unusual for an enzyme to do one thing in one cellular context and something very different in another. All of this we learned by looking at the northern blot for expression. And this helps us to know that the expression is broad. Remember our checklist, BACE, or the candidate clone of beta secretase, should have broad expression, and it does. It also should have broad expression in the brain, because we know that in the brain, amyloid beta can be produced by just about every type of neuron. So here we're taking the mouse tissue, and instead of isolating different organs in the periphery, we take the brain and then we microdissect it into these different regions. And then we run the gels, do the transfer, do the probing, and see, wow, look at all that broad expression. Everywhere we look in the brain, we see those 3 major alternative splice variants of the BACE transcript. So that's wonderful. And finally, we can use that cDNA to make a peptide as an antigen. So go from cDNA sequence to synthesize a little peptide based on part of that sequence, and then inject that peptide into an animal to make antibodies. And then we can use that antibody in a Western blot application. And here they've got 3 sources. The 293 cell line itself, they extract protein, run it on gels, and then do the Western blot. They detect BACE there at the predicted molecular weight. And then here are here is tissue from a control patient and tissue from an Alzheimer's patient. And we see the protein at the right molecular weight in both the cell line it was cloned from, a control human, and an Alzheimer's disease human. So again, everything we would expect to see with this protein and its expression is seen. And I don't want you to worry too much about what these things are, the O, S, and A. The one we care about is the serum lane, the S lane. The other two are controls. Okay, so let's not worry too much about those right now. All right, folks, that's the end of part 1. So I'll take a short break. I'll come back for part 2. In part 2, we're going to go through the rest of the figures, but first we're going to reintroduce the idea of subcellular localization of proteolysis of APP. And then we'll take a look at the more biochemical side of this paper, where they're describing the protein's localization in cells and then its proteolysis. Okay, back in a few.